session. And our first speaker is Professor Maria Kurnikova. Yes, from Carnegie Mellon University. So Maria did her studies in Russia, if I'm correct, MSc and PhD in theoretical chemistry, University of Pittsburgh, and then she moved a little bit to Tel Aviv, Israel, as a research fellow, and finally settled in Carnegie, taking up various positions from 2003, assistant professor, and since 2009 she has been associate professor in chemistry. So I welcome you to the talk. Her research expertise is in computational chemistry and biophysics. Right. And so today it's about ion channels, models for ion channels.
Thank you for the nice introduction. Good morning, everyone. Does mic picks up right? Well, I'm very new to the community. Thank you very much, organizers, to inviting me. This was a very interesting workshop, despite language differences. But I did learn a lot, and yeah, I don't know who found me, but I'm very grateful for having chance to present you some of my work. I'm kind of on the borderline with the systems and equations which I'm using to model in terms of applicability. So there is a very nice sizable crowd of people using continuum theories, that is PD equations, for systems of macro to mesoscopic size in my kind of line of thinking. And what I've been doing, we were bringing them closer to molecular scales, where applicability of some of these methods become questionable. So every time I apply a model, I need to justify very heavily whether the model will be actually applicable, and I have to actually modify models to make them applicable. The reason, well, I'll show my reasonings to use these models, but this is exactly the size scales and the time scales which of the systems which we're trying to model are exactly borderline. They're very huge molecules, so this is one of the pictures. I'll show you more pictures. The big molecules, molecular complexes, they're a little bit too heavy to simulate as chemists do, and they're not quite as big to apply continuum approaches to them in a just straightforward manner, because they're exactly borderline systems. So between micro and meso scales, and that's what makes them difficult, but they are also extremely important in biology, so one need to have some approaches to bridge the scales. So I'm truly trying to bridge the scales by bringing together methods of computational chemistry and methods of more continuum level material type modeling. So yeah, I didn't elaborate title which I put with the abstract, but this is basically the essence what we're trying to do. This is at least the topic of my talk today, is trying to model ion permeations through biological ion channels. Those are well, most of people here, despite being mathematicians, are familiar with structure, some substructures on the cell. So if this is a little piece of, oops, sorry, didn't mean to do that. That went far. How do I make the, okay. So if this is the membrane, that is a chunk of a membrane of the cell, most of you know that it's populated, heavily populated with proteins. Quite often on the mitochondria, that probably 70% of the volume will be protein actually. In some cells, there is a little bit more lipids, but there are plenty of proteins in the membrane. And some of them span the membrane like this. And those, again, subclass of these forms pores. And so that's what I will be looking at. So they are proteins which insert in the membrane, and they create channels for water permeation and permeation of charged and polar species, ions and polar molecules. Well, there are channels of this sort are found in all kinds of cells. They're especially important in active cells, for example, in the heart or the brain, in the neurons, because they are the structures which allow for electrical activity of the tissues. In part, so they participate in action potentials, synaptic potentials. We've seen a talk yesterday about structure of the synapse and the inhibitory glycine and GABA receptors. So those are big proteins which also have the ion channel domain. That's their function. And therefore, since they sit on the membrane, they are targets of lots of drugs, because once you drop a drug near the cell, the first thing which it will encounter are membrane proteins, because that's where the outside of the cell is, and therefore nearly 50% of all drugs actually target membrane proteins, all kinds of receptors and ion channels. So these are very important in pharmacology. So understanding of these structures is very important. However, it's extremely difficult for structural molecular biologists to decipher the actual molecular structure of these proteins. Membrane proteins is a class in its own, and they're extremely difficult to crystallize, to find out their structure. If you would like to do rational drug design, you would want to know the structure, because you want to know how to shape the inhibitors and molecules which will bind to it. That's one thing. And so in the absence of the structure, the major method which electrophysiologists or molecular pharmacologists use to characterize these proteins is actually measuring current. So that's the one and only way to figure out which channel you're dealing with, is to apply voltage across the membrane and measure current through it. These structures are, they are molecular size, so the channels they form are extremely tiny, and they're very specifically shaped, and therefore the current characteristics for each of these channels are extremely specific. So they are like a personal signature of each channel. By measuring the current, you can figure out which channels you're dealing with. And when they malfunction, they change their current characteristics. And that's how lots of pharmacology works, by basically trying to add some chemicals to the cell or to the patch of the membrane and measuring currents to figure out whether the effect of the drug exists. So the ion currents, so computing ion currents would be extremely important, because if I can take the structure and compute ion current, I could relate directly to the experiment, which is the only experiment. That's basically the nearest, closest microscopic experiment which is done, is to measure the current. So until I can predict the current, I cannot really directly compare it to experiment. So in that respect, trying to compute the current through the open channel is a very important problem. Okay. Right. So they come, as I said, in various shapes and sizes. This is one example of a really big and simple and straightforward ion channel as far as molecular ion channels are concerned. So this is alpha hemolysin. It's a protein which is secreted by bacteria, and they are toxins. Or what they do, they, oh, sorry. went. Slight push of a button, everything is changing. So this is, I see, sorry. It's too early in the morning. Okay. Alpha hemolysin. So this depicts a true atomistic structure of the protein. So it's a simplified picture, but in principle, for this structure, there is an X-ray highly resolved structure, and position of every atom is known. So this is fairly simple shape. It's fairly rigid. So once it inserts in the membrane, it passes all kinds of water and ions, whatever can go through. It forms a tube, basically. Almost whatever can go through will go through. The sizes here, membrane is about 30 angstroms. We're measuring distances in angstroms, which are 10 to the minus 10 meters. And so this is about 30 angstroms, and the diameter of the spore is about 10 angstroms maybe. So it's fairly large by atomistic standards. As I said, it's a toxin, so it will, it's secreted by bacteria, it comes to the membrane, it inserts itself in the membrane of eukaryotic cell, that is our cells, and because it's such a big tube on molecular scale, it will let ions in and out in the cell. And basically, equilibrium of ionic concentrations in the cell means death. So it kills the cell by allowing free transport, which normally is not allowed on the cell. Normally, the ion transport is heavily regulated by the cell. So you have to have this difference in the concentrations of, for example, lots of sodium outside, lots of potassium inside. As long as you open the pore and they equilibrate, you're dead, because you lose this membrane potential and lots of other things. So this is how toxins kill. Okay, that's one example. This is, because it's such a robust protein, it also was used heavily in all kinds of bioengineering. People send, it can thread a piece of DNA through this. There were some sort of sensors designed when you attach other molecules to it, and you can see the permeation of polymer molecules through it by capturing it on the other side. So these are all artificial experiments which were done using this alpha hemolysin channel, because it's bacterial, it's very robust. It's a nice little tool for creating pores in the membranes. And if you want a very regular microscopic pores, protein is one of the good examples, as long as it's stable. Okay, so that's motivation. Okay, so in terms of modeling, so I look at the problem more or less as a physicist. So I'll simplify it to death and make the simplest possible model, and then I will bring back complexity when the simplest models don't work. So what is a membrane and a membrane channel from the point of view of very, very basic physics? So the important part is that lipid bilayer, the cell membrane, is made from these molecules which insulate water in two compartments, inside the cell and outside the cell. So this layer of only 30 angstroms of lipid bilayer basically works as an oily film on the surface of the cell. So you have water, and then you have this droplet of oil, and then water again. Why did I call it oil? Because this is a very low dielectric environment. So it's a really dielectric. It's like insulator on the wire for people who are not really physicists here. So you have the current inside this, which goes through the metal. But on the surface, you separate it from the air and other metals by the thin sheet of dielectric. So this is what the main basic function of the lipid bilayer, of the membrane surface is, to insulate internal components electrically from the outside. Because inside the cell and outside the cell, the blood and everything, all the liquids are electrolytes. So if you apply electrodes to them, they will conduct current. Water is a very polar environment, and there are lots of simple ions going on there. So you actually live in the media, so we consist of the media which is very, very highly conductive. So in order to separate these conductive compartments, that's what lipid does. It's extremely efficient. So if you compute the energy of taking a single ion like sodium and try to drag it through the lipids, through this very tiny 40 angstrom thickness of the oily film, it costs something like 20 kilocalories per mole. So it's a huge barrier by the standards of chemistry. Well, I don't know. I mean, some of people probably do not used to think in terms of energy. So anything. So the probability of something happening in biology is more or less governed by the Boltzmann distribution of energy. So if this relates to temperature, but RT is basically if I measure energy in terms of RTs. So RT at room temperature, if I have room temperature, which is about 300 Kelvin, then RT is approximately 1 kT half cal per mole. R is times Avogadro number, so we leave the Avogadro number out. But then the unit measure is about, let me think, half kcal. Run out of water. Okay, wasn't very, okay. RT. Let's do per molecule, KT. That will make life easier. KT is approximately 0.5 kcal per mole. So usually all biology talks in terms of kcal per mole. Well, some people in Europe do kilojoules. Joules is 4 times smaller. So this is my unit measure of energy. And the probabilities go by Boltzmann, the exponential. So if I change energy here, the difference 1 kcal per mole will give me 10 times difference in probability. So if something, if I have energy barrier which is on the order of 2 kcal per mole, or let's say more than 2 kcal per mole, let's say 5 kcal per mole, it's already a fairly stable thing. It will happen with very low probability. So if I have a barrier through the membrane about 20 kcal per mole, then I can safely say, oh, sorry. Then I can safely say that ions will not go through. It's a huge barrier. 20 kcal is really a huge barrier. So several kcal per mole creates already a barrier for the process to happen. Okay, so this is as far as actual physics of the problem is concerned. I don't know why this thing does. Yes. Okay, so in order to build a model, what do we need? We need, so basically, we need to account that the dielectric barrier through the membrane prevents direct permeation of ions through the membrane. So I can separate compartments in the model, and I know how much it costs to go through this layer. I need to set concentrations of ions outside and inside the cell differently, because they need to be a gradient, because that's the life. And then because I have the different concentration of ions and different charges on the ions, by the time I count all the ions, I have the actual electrical potential, net electrical potential across the membrane. Another way to think about the membrane is an analog of a capacitor. So I have two metal plates separated by the dielectric. If I have some sort of charge accumulation, I would have the voltage on this capacitor. So if I put it there, on this. We'll see how strong that is. Okay, I'll try that. Right. So basically, there is a net voltage which is created across the membrane by all components at once. So some of the lipid has charge, something happens outside elsewhere, which brings in electric potential. So we cannot really, I think that's what freaks me out when she changes the look of the screen. Then I change the slide. So lots of things create this net potential in the membrane. So from the point of view of model, one can safely assume that there is a certain measurable voltage across the membrane which is created by environment, and that will be different in different cells. And for example, in the neuron, at rest, when neuron kind of sits there, does nothing, this voltage across the membrane is about, so across right there, it's about 60 millivolts net voltage. And so when you start permitting, you open the channel and start allowing for the current to go through, this voltage may change locally. And that's actually how neurons work. They change voltage across the membrane locally. They create a certain short circuit, and that's how it works. Okay, so that's the simplest, roughest model description which there is. And this fits, now see, hopefully these are just examples of other structures, just how various kinds of channels look like. This one I made in roughly all atoms have proportional size to what they are, and this is the size of the pore, more or less, what you're looking through the pore there. So this is the actual structure of the channel. That's another example of a channel which is selective, and I'm not even going to go there today. The selectivity to specific ions. Some of the channels conduct only specific kinds of ions, and that's chemistry, more or less. Right, so my model approaches, as I said, starting with the description which I just spent 15 minutes doing, fits very nicely in the material type, continuum type, partial differential equation type descriptions, which is in this business called Poisson-Nernst-Planck, which combines PD for electrostatic Poisson, that's everyone recognizes, and Nernst-Planck is basically drift diffusion. That is diffusion with, I think yesterday people called it convection term. We call it drift because that's the driving force is created by electrical potential, and the ions go because they're charged. So that's the continuum description, and that will go directly onto what I was hand waving my way through right now into that model. But as I just showed, these structures really very tiny, and some parts of these proteins, so these yellow balls there are ions. So that's a selectivity filter. This is where protein actually kind of clumps to the ion, binds it a little bit, and senses what kind of ion is it. So if it's a wrong kind of ion, it will not properly fit, bind, or whatever, and it will not go through. So that's the selection. And as you can see, comparing to the protein size, these ions are huge. That is, this is no longer a tube which conducts liquid. These are really particle by particle level permeation at this stage. So the model which uses PDEs as a drift diffusion becomes questionable at this level. So the next better model would be to perform atomistic simulations, what's called molecular dynamics, basically describe every atom in the system. The simplest level, simplest approach is to write Newton's equation, that is, think of them as classical particles and solve their motion in the environment. Unfortunately, that's extremely expensive, so I can run on a big computer, easily run, let's say, 100 nanoseconds of simulations, and a single ion might take 50 nanoseconds to permeate, which means to collect the currents, I need lots of events that will take forever on the computer. I'm talking months, not really hours or days in the simulations. So it's prohibitively, it very quickly becomes prohibitively expensive. So there are some bridging models where you think of ions as diffusing species in the liquid, and you can design a scheme like Brownian dynamics scheme or Chmielewski type equation, Langevin type dynamics, and treat ions as particles and water as continuum, which is a mixed model, and again partially applicable but not in every situation, because it already approximates water, which is as a continuum on the but the water molecules is on the order of the size of the ion. They're about the same size, the water and an ion. So if I treat water as a continuum, it's a huge approximation. Okay, anyways, that has been done. In any case, let's see. This is the simplest way to write the equations for this. So this is diffusion. Yes?
You said that this molecular dynamics simulation is out of reach, but I thought Benoit Roux was doing this kind of thing.
Yes, Benoit Roux, Verdegroot, like there are. I've been doing this. There has been 2 or 3 examples where currents were calculated directly from MD simulations. They're fairly exotic. Actually, Benoit doesn't do that. Shaw Corporation did some microsecond simulations where they directly compute current. Even for the small channel, you can do it once by employing a huge computer for yourself for a year, and then it's a showcase paper in Nature or Science. So you cannot really do it routinely when you collaborate with a pharmacologist yet. And even there, they cannot do real current voltage characteristics. They would do 1 or 2, 3 points on a current voltage curve. I'll show you some MD. We run MD, so I'll show you how it works. Yes, you can run MD in principle. You will run into all kinds of other problems. How to apply voltage, how to set concentration gradients, how many species you can have. And then once you bring in chemical quality of modeling, you also will quickly run into problems of how good the ionic representation on the molecular dynamic level. And then could you trust your results from MD simulations, because your parameterization is not ideal, and it should be not really current MD, but rather polarizable, and people from quantum chemistry will come in as just exactly the same kind of worms. So once you start going into atomistic things, a quantum chemist will come in and say that you're doing all wrong, and ions are really complicated species. And so MD is not the full solution to the problem, even if you can run it. But yeah, that's more or less the best at the moment what you can do. What Roux is usually publishing, and we have some examples of doing similar things, is trying to compute selectivity, difference in energy for the particular position of ion in the channel. So you're not trying to get the current, you're rather trying to just calculate how much does it cost to bring an ion from solution to a specific point in the channel. I'll show you some of these calculations. Okay, so continuum approach to get the current. Current is, well, the idea to model current as a diffusive process is not crazy, because diffusion equation is very general. So as long as I know how to set up the energy profile, diffusion equation is applicable all the way to microscopic scales. So in this case, the current or flux I wrote. Basically, I'm going to solve for steady state. Divergence of flux equals 0. Flux is the created by the concentration gradient with certain diffusion coefficient, which will be space dependent, and drift, which is the depends on the concentration of the mobile ions, C. The Q is the charge. So because these mobile ions are charged, they are driven by the gradient of electric potential phi, which is the subject of the Poisson equation. So phi is the electrostatic potential. This is the Poisson equation. Epsilon is the polarizability of the environment. So epsilon is going to be a high, like this is my cartoon of a channel. If this is the membrane and this is the channel, and this is water, then epsilon B will be high, like dielectric constant of water is 80, and epsilon in the membrane will be low. Dielectric constant of the membrane as the dielectric is 2. So it's 2 and 80, more or less jump here in this picture. And the sources of charge in the Poisson equation are partial charges on protein atoms, because the protein structure itself will create some distribution of charge in each atom, and that creates certain electric potential in the environment. This is fixed, it's preset. And then, of course, electrostatic potential is created also by the mobile ions which redistribute. So I end up with a set of linear PDEs. I have to write the diffusion equation for each species. So I stands for the species of the ions. For example, sodium chloride, I will have to have sodium, and I will have to have chlorides here. Different diffusion coefficients, different charge, different sizes. So two fluxes, and coupled to to the Poisson equation. We usually solve with Dirichlet boundary conditions, setting up electric potential somewhere far away from the channel on the boundaries of the box. Could be set to 0. This is not very good approximation because it's not far enough. So usually set to be pre-computed value based on some simple calculation, Coulomb's law or something like that. And the boundary conditions for salt is usually, again, preset concentrations on the boundaries meant to represent the bulk solution, salt solution concentration. Like, for example, yes?
What do you know about the dielectric constant inside the channel? I mean, do you assume it's uniform? Is there any good reason to do so?
The channel, dielectric on the channel is a tricky business. That's where this approach starts breaking down. So epsilon is not very well defined for the protein. People are using, I usually use around 4. It's slightly more polarizable than the membrane. For different purposes, in the literature, people use anywhere from 2 to almost 30. Well, let's say if you want to calculate protonation, you set it to 10. So it's a not very well defined thing, because protein is locally polarizable, and if you're dealing with, so it depends on the model a little bit. If you're dealing with the rigid protein, you can preset polarizability to more or less match the local polarizability. Or if you actually allow for protein to move in space, then you would like to set epsilon low, because then the rest of the polarizability will be taken care of by fluctuations of the charges on the atoms. So the protein polarizability is slightly complicated question, but well, in this modeling, which I'm going to show, I'll point where it's a tricky thing. Yes?
But 4 is a very low value, because basically the reason the membrane is impermeable to ion is because of this epsilon equal 2. isn't it?
Yes, exactly that.
So 4 is not that much bigger.
There are charges. Yeah, let me show you what do I have on the next slide? So this is right now, oh, okay, so I have a whole set of simple examples. So when I come to protein, I'll answer your question. So in this case, this is just the hole through the wall model to show the, formulation, formulation of the model, but then I'll show the protein and I'll discuss the difficulties.
So just before you continue, just to clarify, so here you have, in terms of ions, you think about having sodium, potassium.
Concentration. Well, in this case, in these calculations, I'm going to have just, two species, one positive, one negative. Let's say sodium and a chloride.
Well, with the same concentration.
Yes, but we're going to, positive and negative will be set to the same concentration, but they could be different in different compartments. So we'll have concentration gradient between the boundaries, but I will always set it to be, well, the same. The same for the positive and negative species at the boundaries.
Okay.
And the selectivity, which kind of species go through the channel, will be determined by the structure of the channel.
I see. And so, and you have reflective boundary condition, I guess, for the flux on this on the membrane, except at the small pore, right? In terms of the flux.
Yeah. So, but that's a limitation on the model. So basically, in theory, I should not have set any boundary conditions except on the boundaries of the box, and everything else should be taken care of by itself. Okay. But in reality, I'll show you where this theory doesn't work in this case, because ions are treated as simple concentrations. They do not really understand dielectric properly. So you have to really prohibit current in the membrane at this level of approach. And then I'll show how to correct for that. Yeah, that's a very good question. Right, so, okay, so this is the simplest, sure.
Out of curiosity, you mentioned that there is a 20 kcal barrier to pass through the membrane, and generally people doing MD was able at least to compute the profile of the free energy barrier that an ion see when passing through the channel, right?
Well, that's the channel.
Inside the channel.
That's the specific.
So I guess you have an order of magnitude, an idea of this barrier, how they decrease through the gating process of the channel, right?
Yes.
So this maybe can help to set up the level of the electric constant, as was discussed before, even if the electric constant is ill-defined at this length scale, right?
Yes and no. So in this, in this model, this is the extreme case of the ion channel. Right, so I have a membrane, low dielectric, and I have a water-filled pore. I have only two dielectric kinds, so in principle, this is flat. That is, whatever fits in the hole should be able to go through. So right now, I have a big barrier on the membrane and a zero in the pore. So that's the limit, extreme limit of the model. In reality, you have profile which is formed by the protein through the pore, but it's a low energy profile. Now, the problem with this model, well, I'll show you on the next slide, that if you set the equations like this, nothing prevents the positive and the negative ions to, they're infinitesimally small. They neutralize each other. They can sit on top. Well, there is no size of ion here. It's just a concentration. So any volume in space can be filled by the concentrations of both positive and negative ions. Then the volume in space becomes neutral. And then it's insensitive to dielectric. And therefore, if you set the equation like this and do not prevent ions from going in the membrane physically, they will go through. And I hope this is right on my next slide. The next slide, it's a departure into particle kind of simulations to set up the comparisons. Do I have that? Yeah, that's, no, wait a second, too far. Yeah, here it is. So that's an example how continuum dielectric models doesn't work for this situation. So this is an example where I have the membrane, no hole. So that is dielectric equals 2, dielectric 80 on two sides, equal concentrations of ions set to 0.1 molar on both sides, positive and negative ions in the simulations. That's purely PNP model. And the black dash is the distribution of ions as a result of this solution. That is, they neutralize each other and they happily go in the membrane. They don't see the membrane. That's Poisson Nernst Planck at the microscopic scale without boundaries for the membrane. Okay, now if you do, as my previous skipped 5 slides, we're doing the, if you take Brownian dynamics, in this case we run actually dynamic Monte Carlo on the grid to run it simpler and faster, but it's equivalent to Brownian dynamics. So if I do Brownian dynamics, that is, I still have dielectric distribution and my Poisson equation is the same exactly. But instead of doing diffusion equation for the ions, I am actually counting particles. So now ions are particles, they have size, they have radius, they're hard spheres, and water is still no viscosity, nothing, so it's just a dielectric continuum. So water is purely represented by a dielectric continuum. So if I run a particle simulation like that, then they cannot neutralize each other, so they do see the membrane, they will not go in the membrane. So if I have a barrier, so I'll skip the formulation, so it's a liquid junction. Let me skip through all of this. So if I have a barrier, where was that slide? There, yes. Then the blue line is for the results of the dynamic Monte Carlo or Brownian dynamics. So then basically you have bulk concentration of the ions set far away outside of the membrane. The density of the ions depletes going towards the membrane and no ions go in the membrane. So there is no special protection boundary on the membrane in this simulation. It's purely electrostatic effect. So this effect is due to so-called self-energy of ion when it goes in the membrane. I'm not showing the energy for the ion going through the lipid bilayer here, but what I'm going to compute is the self-energy of the ion, green one, going through just the cylindrical tube.
What is the length scale in the previous plots?
Length.
The length scale of the decay of the
Good
what
question.
What is the length scale of the decay of the concentration?
So this is about, when it starts decaying, like 15 angstroms.
I can see what it is, but what sets it? What
The thickness of the membrane.
No. Really?
Yeah. Thickness of the membrane is about...
No. I'm asking the length scale of the sort of exponential decay of the concentration near the
it's Debye length, basically.
Debye. Okay.
Yeah.
Okay.
It depends on the concentration and...
What is the size of an ion?
About 1 angstrom.
Sorry?
About 1 angstrom. Yeah. The size of it, it's a Born salvation energy, basically. The size of the ion is 1 over, so the difference in energies is 1 over 2a, where a is the diameter or radius of the ion. And 1 over epsilon is the, difference in the epsilon, mostly size of the ion, I guess. And the concentration will come in, so it's a Debye length. Right. So, taking a cylinder to make a small hole, I'm going to compute the energy. Why does it do this? The energy of ion going. So basically, I take an ion from the bulk and take it as a single particle through the different positions in the channel through the center of the channel. And the channel is this simple cylinder. The radius of the cylinder is 4 angstroms, so it's a medium size as far as the ion channels are concerned. And if I compute this energy for the positive ion, then I have for the ion going through the neutral pore of about 4 angstroms, I get about 2K. Well, it's in KT, so it's a 1 kcal per mole barrier. So just the presence of the membrane outside the hole creates this barrier already. So this is what is missing from the PNP formulation. This is ion self-energy. This comes from the size. It's Born energy, basically.
Is this an entropic barrier or
Purely both, because it's a Born energy. Part of it is the entropy of creating of the hole, the cavity for the salvation. But it's clumped in. So at this level, this is a purely energy, potential energy. It's electrostatic potential energy, basically. But if you think in terms of the degrees of freedom of water, then it's a free energy. So we call it free energy so that we can easily migrate between micro and continuum theories.
And so, how did you calculate this? How did you make this? This
is purely Poisson equation.
So you solve Poisson?
So I take a static ion. Yeah, I solve Poisson for difference in energies for each position of ion along the center of the pore, and I plot this energy, and I set it to 0 away in the bulk. So basically, I'm comparing energy of the ion with respect to ion being solvated, happily solvated in bulk water, and somewhere in the channel. This is the center of the channel. So
there's also a contribution from the translational entropy of the ion itself. Do you know if it's much smaller?
You lose some. Well, it's 2 to 3 degrees, right? So in the bulk water, it has 3 degrees of motion. It's a tiny particle, so it's 3 degrees of motion. In the channel, it becomes more like confined. It's not purely one-dimensional, but of course confined. So you lose some degrees of freedom there. It's not huge. It's units of kcal per mole. Oh, sorry, units of calories per mole, this entropy. But I didn't take it into account. One can easily add it in there.
It's the same order.
No, this is kilocalories per mole, and that entropy.
Forget what calories. I don't know what that is. But
No. A kilo. You know what kilo is? It's 10 to the 3, so it's not the same order.
just to get your scale, and you've got basically an energy barrier of 1.5 kT.
Yes, which is half kcal per mole, and kcal.
KBT. So this is 1.5 kBT, which is also the entropic cost of going from 3 to 1.
No, you're not going completely 3 to 1. You confine, so you can compute it, but it's not full kT. You're not losing that much. I mean, this can be accounted for. That's the, let's say, easy term. Right. Okay, so now if I go back to PNP and compute currents, then this is current voltage characteristic for this simple cylindrical channel, which I showed as a picture. And so this is the black line is the original, the current predicted by pure PNP, and the values are on the left-hand side. So it's 4 picoamperes, 40 picoamperes for, let's say that's my maximum, 300 millivolts. And the blue curve is from dynamic Monte Carlo. That's particle simulation, continuum diffusion of particles. So, and that's like 10, basically, at the extreme, 10 times smaller. So that's the result of that 2 kT barrier, which is really very small. The PNP can be amended by this self-energy, so it's not the end of the story. If one adds, I'll show the formulas. So I can do, so this is formulation which I showed before, PNP. And this is where I add this dielectric self-energy of the ion back into continuum equations. That gives the curve for the current, which more or less brings it back to the Monte Carlo result. So this is now comparison of the generalized, let's say, let me call it generalized PNP, compares well with Monte Carlo. And, oh, sorry. But, okay, this is comparison of the ion distributions through the channel center, just one line, center of the pore. The PNP, it's a little bit messy. So the, let's say, for the anions, pink curve is the generalized PNP. And the light blue or cyan curve is the Monte Carlo simulation, which was clearly not sampling a lot, but sufficient to show the shape. The black curve with dots was the original PNP result. So the distribution of ions is off too. Okay, so if I take a slightly more realistic structure that is actual protein, this is the tiniest ion channel in the world called gramidine A. It's really slightly unfair to the model because it's so narrow. But we did use it as a model as other people did. So in this case, I set up dielectric. So the picture through the box shows the colors on this box show the distribution of dielectric constant. So now we have a separate dielectric constant for the protein. It's still set to be low. In addition to the dielectric constant, I don't have a picture. I have a picture somewhere further down the presentation without the channel where it shows charge distribution. But basically, in addition to just dielectric on the protein, each atom has a partial charge. So even if the protein molecule is neutral overall, it has lots of dipoles and quadrupoles and everything. So because of the charge distribution, so it creates electric field. I'll show the picture. It creates electric field in the channel which is conducive to ions, and that's how the spore is made permeable. The spore is so tiny that if you leave it neutral, just a hole in the dielectric, there will be pretty much no current through it. And so basically, if I compute the same thing, compute dielectric self-energy for the ion through this channel with standard parameters, epsilons 2 and 80, this is the black curve. So the barrier is really very big. It's almost like going through the bilayer. Just because the channel is really very narrow. And the dielectric, so the model is fairly insensitive to the dielectric constant inside the channel, again, just because it's so small. It will be fairly sensitive to the dielectric constant on the membrane, in the protein, which I was playing with, trying to get this curve down. So I can set up somewhat crazy dielectric constant 30 on the protein, and then I get, let's say, 10 kcal per mole. Still a barrier. So in the absence of the partial charges, this thing will give a barrier anyway. So this is dielectric self-energy. So we didn't even try to calculate currents through this. This will not be conductive, no matter what. But then let me skip through. One realizes that in reality, there is an adjustment between the protein and the ion, even in the narrowest part where ion pretty much binds into the channel, shading all the water, especially there. The channel atoms will adjust to the presence of the ion. So basically, you're taking the ion, which is positively or negatively charged, it's a big charge, and bringing it very close to other atoms which all have partial charges. So all the dipoles are going to orient towards the ion. That's taken care of, should be taken care of by the epsilon, but could be taken care of by MD simulation. That is, let protein relax to the presence of the ion. And that will create certain polarizability, which is better reproducing actual protein polarizability than just a dielectric constant in a rigid protein. So protein is not rigid. So we did some very small MD simulations. And this is what happened. So my simulations are really short in this case, less than nanosecond. They just to position the ion in a specific point in the channel and let the environment relax. That happens very quickly because the adjustments are really very small in terms of the repositioning of the ions. And this is still a very simple model. And this is what it does to the self-energy of the ion. So if I compute now dielectric self-energy of the ion through these relaxed channels as an average, this is what I get. So this is the original curve with the rigid structure of the protein, and this is a curve which I'm getting from the relaxed protein. This is the energy for the positive ion, and this is the energy for the negative ion. So in addition, what I got as a bonus is the selectivity that this channel is really preferring positively charged ion. That is, it will be conductive. Here you can see the energy is pretty much wiggling around near zero, and that creates the hole which will be conductive. In this case, this is still a barrier for the chloride, so it will not go through. One can recompute, so this didn't show the current. So one can recompute the repositioning of the ions. They're really not very big, so this is just the fluctuations on the protein, which are created by the disturbance on the protein, which is created by the presence of the ion. So we basically can amend the PNP results with this pre-calculated free energy of the ion. We can definitely do a better job. So I'm going to switch the channels to a bigger one. But so we can do a better job on computing the single ion interacting with the protein. And this is what a reasonably large field of molecular simulations of ion channels have been doing for many years. So it's getting better and better in terms of I can compute potential of mean force, which is the dielectric self-energy with relaxation of the ion going through the channel. Then I can put it back in the equation. I think I lost the equations somewhere. I think I skipped through them. Oh, no, they will come later. I just did the presentation in odd order. Okay, so just to illustrate the hand waving which I did before about the ion protein charges. So red and blue dots on this protein are the partial charges. So this is the distribution of, this is rho fixed in the Poisson equation. So even if the protein is neutral, this will create a certain electric field, inhomogeneous electric field in the medium, and that's what makes it kind of difficult. So this is back to alpha hemolysing, the very robust and fairly big protein, so where one would think that PNP theory should work much better, they're more applicable. So we have now the equation of diffusion has been slightly modified. In the original PNP version, I had just phi here, which comes from the Poisson equation. Now it's a psi. It can be, it can be as simple or complicated as one wishes to. It depends on the model a bit. But basically, the Q times phi is the original term, the original PNP formulation. I could add, so when we were discussing that I have to set the flux to be zero through the membrane in the original PNP formulation, I could put this condition as an additional term to this psi. So I can put it as a boundary conditions, or I can put it directly in the equation. So this is where this is poor man's ion self-energy goes in the case when you do not really calculate it, but say ions don't go in the membrane. So this is W then for hard kind of spheres type or hard membrane type approximation. Now, when I compute dielectric self-energy by doing separate Poisson calculation for the ion and single ion energetics, this is where I put this energy term. So this will be ion dielectric self-energy, or I can take full-scale atomistic simulation and compute free energy of true free energy of ion going from the bulk to the protein, taking into account loss of entropy and entropy of water and creation of the cavity in the protein instead of in the bulk water, polarization of the protein. If I'm very sophisticated, I can switch models and have all the atoms polarizable so that they correctly respond to electric presence of electric field. So this could be all done as a kind of first layer of the hierarchy on the completely atomistic level. Then out of my atomistic simulation will come this psi energy, which can go directly into the diffusion equation. And then it still needs to be solved self-consistently with the Poisson equation, because distribution of electric potential changes as things move. So the only thing which the generalization which we actually did does not include is on-the-fly adjustment of the protein, because we're pre-computing phi for the single ion in the absence of all other charge species and in the absence of the applied voltage. So this is where self-consistency is somewhat broken. So if I could bring in back flexibility of the protein into the model, that would be more self-consistent. But at the moment, this is pre-computed for the single particle. And then the influence of other charged atoms are still added on the mean field level via Poisson equation. So that's the modification. Then what we discovered is that just solving... Can I take like 3 more minutes? 5 minutes, okay. Right, so just solving Poisson equation turns out to be in 3 dimensions. Turns out to be difficult because of this distribution. This is also governs the ionic distribution. So concentration of ions will be exponentially, by Boltzmann, exponentially dependent on the electric field. And despite the fact that both diffusion equation and the Poisson equations are linear equations and easy to solve on any kinds of grids, finite elements, anything, the coupling between the two are exponential. And so this exponential becomes very large, very fast when 2 charges come together very close. So every time I have a charge on the protein and the ion goes into this really narrow space close to the atom of the protein, in reality, quite often these particles interact, the particles of the protein and the incoming ion interact very happily. That's what makes recognition sites and binding sites work. But in the PNP formulation, this creates instabilities. That creates very big gradients at the boundaries of the dielectric jump between the protein and the channel dielectrics. So so if you have the boundary and you have the charge right next to it, and you have an incoming charge, then there are very big gradients, phi gradients, and that creates instability, and it grows exponentially. So to actually solve this system of equations, we used very severe under-relaxation. That is, you kind of relax the equations by fractions of steps and iterate between the equations very often. Because if you converge one equation, then the second one will blow up immediately because of this coupling between the two. So we kind of struggled with the numerical methods for a while with this system. But then we realized this is really should not be happening that often, because in reality, atoms adjust their position. And in real molecular systems, there are no huge electrostatic field gradients. The incoming ion will just never approach the very big gradient. So it will kind of stop just because atoms have fairly rigid local interaction with each other. So if one takes into account the fact that the real protein surface, which creates the channel, is really soft. That is, it's not a hard wall, but consists of atoms which are soft. So this is a picture illustration. I don't know. I don't want to really. Yeah. So if you have a charge nearby the surface, then the opposite charge will attract, try to go in the prohibited space, and blows up your equation. In real molecular system, that doesn't happen. So it's just an artifact of hard boundaries. So we amended this. Let me skip through all this really simplified ion. So this is an example of a group which may line the surface of the protein. So if you look at it at really microscopic level, it's not a hard surface, but rather collection of atoms, which in this picture, sorry. Where did it go? Yeah, in this picture, these are positive ions, this is atoms, these are negative atoms, and this is a kind of dangling end of lysine of a group which can swing, has a partially positive charge on the end, and it will be on the surface of the ball and can swing up and down with the electric potential or the flow or incoming ions very easily, so there is this adjustment. So what we did, we went back into molecular dynamics simulations and pre-computed this single ion interaction with various protein groups which may line the pore. On the molecular level, that creates a much softer function. It's potential of mean force, or double correlation function, which can be easily computed from the simulations. And it looks, then we just fit it. And it looks very softly. So this is now the ion approaching the wall. This is how it really behaves. It's not a hard wall. It kind of comes in and then bumps into it. Rather, a soft approach for the positive ion. This is the approach for the negative ion. So if I use that PMF to put back into my generalized PMF model, so these are the problematic areas actually on the protein. These are where protein has some charge exposed to the channel, and at the same time the channel is narrower here. So this is where the equation would blow up standard PMP formulation in using standard relaxation methods. And so that, let me skip through the diffusion coefficient stuff. I have 5 minutes. Yeah, that's the result. More or less. So this is what all it does. The softening up of the formulation of the protein wall creates this. So this is a two-dimensional slice of the calculated current field or flux. For red is for chloride, and blues are for potassium. And this grid is shown only for this region in the protein. So the rest of the channel is kind of very simple. The green boundary through the grid here shows the boundary of the kind of standard hard wall formulation of the channel wall, and green line will be more or less the line separation of epsilon. This will be set to epsilon of the protein, and this is set to the protein in the channel. So as you can see now with the soft wall formulation, some current is allowed along the surface of the protein, and this is due to presence. So lysine is the positively charged group, which I showed before, and that's why negative ions prefer to go there. So if I were to plot the distribution of ions, I would see an increase of the negative ion concentration there. So it results in the flux also. So this creates slight modifications on the total current calculated, not very big. As you could see, it's really a small addition. The current is the kind of total through the area. It does create effect on selectivity, predicted selectivity of the channel. That is, whether it prefers positive or negative ions. And it does create effects on rectification. That is, at what voltage the current will stop. So it changes the shape of the current voltage curve. So it's a very kind of minor modification, but it creates several things. So it basically changes some predictions, and it creates much more stably behaved set of equations. So we kind of get rid of instabilities there. Okay, so that's the modifications. So let me skip through calculations of energetics. I don't really need any of that. How you position the protein in the membrane also influences the resulting currents. Let me, I just want to, yeah. So this is the current voltage characteristics calculated. The colored lines are various experiments. As you can see, there is no exact consensus, but all predicted. So this is purely PNP amended only by the ion interacting with the wall. So I don't have self-energy there, so PNP overestimates the results, but I wanted to show that the shape of the curve changes. And let me jump to the end of this. Probably the easiest way to do it. There must be an easier way to jump to the end. So, right. That's kind of a summary of the computed properties, electric properties of the. Why does it? Now I jumped out of the presentation. Right? So I'm not showing total currents. Total currents don't change that much, but this is the prediction of at what voltage current stops through the membrane, through the protein. So the black dot is the pure PNP. And then with all the corrections on the membrane positioning and taking into account the protein relaxation, everything except for the ion self-energy there. This level line is the experimental line. So we kind of, with our calculations, we undershoot and overshoot, but much less than the original PNP, which is kind of completely off. And with that, I skip through the conclusions, which there are plenty, and I have to acknowledge the contributors. This work was done in collaboration. So mostly what I showed today was done by Nikolai Simakov in my group, and I have to acknowledge financial support, and thank you very much for your attention.
I take a few questions, maybe one or two questions, if there are any. Yes.
I would like to know, with your method, can you, for example, is there some kind of database where you can reproduce, for example, what biologists measure, some voltage current relations? Can you with this reproduce them? And another thing what biology often uses is the Goldman-Hodgkin-Katz equation, including me. How well is actually this equation, if you look at with the kind of your simulation, can you verify them? This is a good approximation or it's a bad approximation?
Well, if you set up linear ramp, so PNP is the modification on that. So you get Hodgkin-Katz equation from setting up linear ramp of the potential. So forget about the presence of the protein, just the liquid junction, and do not do self-consistent between diffusion equation and Poisson. So you kind of have linear ramp for electrostatic, stick it into diffusion equation, and you get the Hodgkin solution. That's basically even more simplified approach. Works from what I know perfectly well for giant axon. That is, when you have truly macroscopic currents, you will get a good agreement with that linear theory. In all channels which I showed, with even all complications and modifications which we did, it barely works. Comparison. So I have some comparisons to experiments. So the alpha hemolyzin basically is one of those which people studied to show that series work because it's so deceptively simple. You kind of get in the ballpark of the order of magnitude, right, with modern theories, including mine. It's very difficult to predict current voltages to the experimental level. Simpler channels like porins, you get more or less this quality which I showed for alpha hemolyzin, but that's about as good as it gets.
Many of those ion channels are mechanosensitive in the sense that they depend on, for instance, gramicidin definitely has been measured, and I mean, I guess many are. I wonder if you have anything to say about this. So you're talking about soft proteins, so I guess those were very small strain at the scale of the whole protein. So do you have anything to say about mechanosensitivity?
Well, that would be, so let's say all the models which I showed are trying to predict the current from the single conformation of the open channel. So once you start changing conditions of the environment and the channel opens, closes, goes to subconductance levels, you have to reshape the molecule first and then compute the current. So from the point of view of the measuring current voltage characteristics, you usually measure them at a single set of conditions. That is, you create the open channel with given pH, given strength, tension, given voltage, and then you measure your electrophysiology. And this is what I'm trying to model here. To start changing the protein shape, let's say, create the tension and wait for the mechanosensitive receptor to open, or bring in glutamate and wait for the glutamate receptor to open the channel. It's a sort of separate problem on a different time scale. So it's a problem of modeling changing protein conformation. Once the channel is open, the current conduction is really fast on the protein modification time scales. So that's like milliseconds as opposed to hundreds of nanoseconds. And therefore, they are just not done on the same model. You want to do it separately.
I'll take the last question.
So I would like to come back to the conditions you put on the simulations. In neuronal cells, at least, depends on the type of cells, but the concentration of chloride is not equal to the concentration of sodium, or the sodium plus potassium. So here there is an important question that's negative charges, at least in terms of carry by ions, by fast ions, is not compensated by the concentration carried by the fast positive ions. Have you tried to look and try to go into this question to look at how you modify your boundary or the conditions, your Dirichlet condition, to see what could be the consequence of that?
Of the charge on the boundary of the box?
Concentration on the left, concentration on the right.
Well, I did the different concentrations, but they always equal for positive and negative.
Suppose they are not equal.
Positive and negative?
Yeah.
Well, that's hard to imagine. Okay, let's say my boundary is...
I'm talking about for ions. It can be compensated by proteins.
Exactly. So basically, right. So once you move a little bit, let's say 100 angstroms from the membrane, you're more or less in bulk conditions. where you should expect charge neutrality by the mixture, but it's a soup of enormous amount of proteins, ions, molecules, polar molecules, all kinds of stuff, right? So they basically, I cannot really, I'm not modeling the specifics of every species in the cell. But if I measure this solution as an electrolyte, if I think of it as an electrolyte, in the bulk I have to assume charge neutrality. And therefore, if I think of the boundaries being far enough, I could think of changing Dirichlet to something else. But if I think that my boundaries are far enough, I set up charge neutrality no matter what. Otherwise, any physical chemist reviewer will kill me. The discrepancy between ion concentration close to the membrane will happen in this model naturally if, for example, you change the composition of the lipid. All I showed today, which I didn't show any variability on the lipid, so my lipid was just the dielectric 2. But different lipids have different dipoles, and some of the lipids are charged, negatively charged, positively, well, mostly negatively charged lipids. But so if you set up your membrane layer to be more electrically active as the real lipid, like set up the dipoles on the membrane, so that will create certain difference in distributions of ions right next to the membrane, and probably will create closer conditions to what you're thinking about. But I would hesitate to set it far away on the boundary of the box because I need the total charge neutrality. Or I need to add some macro things which negative charge. This protein is not neutral. Gramicydin is neutral, but the alpha hemolysin total charge is very big. It's really not neutral. So it creates huge of difference between this ionic kind of concentrations right in the protein, like next to the protein.
I suggest we thank Maria for the nice talk. Thank you. We welcome our pleasure to welcome the next speaker, Professor Dr. Jens Timmer. Yes, it's on. Yeah, okay. So he's a professor of theoretical physics at Freiburg. He got his PhD in 1994 from the University of Freiburg, and he is he's co-director of the Institute of Physics, I understand, and you're also a faculty
Dean of Studies right now.
And exactly.
It's no fun.
And is a faculty member in mathematics and physics, so that's quite impressive. I think to belong to both departments is very nice. He is a group leader in data analysis, modeling of dynamical processes in life sciences. And his research, I understand, is in interdisciplinary application of mathematical methods and analysis of models in biology and medicine. Yeah, so I welcome you.
Yeah, thanks for the nice introduction, and thanks to the organizers for introducing me. So now we come to something completely different. So far, most of the talks were dealing with the forward problem. Yeah, so you had a set of equations, partial differential equations, funny things, and then you looked for methods to solve them. We are dealing mainly with the inverse problem, meaning we have models or candidates of models, set of candidates, and they have parameters, and these parameters are not known. Yeah, and what we are trying in this inverse approach is to estimate the parameters, and since the data, I will show you some in the following, the data are noisy, this then introduces uncertainty in the parameters, uncertainty in the predictions, uncertainty in the model structures, and so on. So therefore, I'm going to talk about uncertainty analysis in systems biology, and what this means. I give you a little kind of historical introduction in a second. So what we are trying to do in the group are two things. We are very closely collaborating with biologists, so every member of the group is married to 1, 2, 3 biologists or clinicians, and we have very specific projects, and what we are doing is, on the one hand side, we try to develop methods for modeling these systems, and then we really do it. Yeah, today I'm going to talk about the methods part. If you're interested in the applications, we can talk about tonight at the dinner over a glass of wine. So I will give you a very, very brief introduction to systems biology, since we are a very diverse kind of community here, and then I'm going to talk about the different types of uncertainty. Uncertainties in parameter estimation, in finding global optima, in parameters, in predictions, and then a little bit also on the model structures, and then finally I will touch briefly some topics that are around this theme. So systems biology started mainly in 2001 or 2003 when the first human genome was sequenced. Yeah, the sequencing the first genome took, I think, roughly 8 years and $3 billion. Yeah, today it's a little bit faster and a little bit cheaper. You can do it over a weekend for $1,000 or something like this. And when the people who put this forward, they very much hyped this by, if we have the sequence of the genome, then we will understand everything that happens in the cell. We will understand and cure every disease, and so on. And of course, this hope did not came true, because it's not in the gene, but the genes produce proteins. Proteins form networks, metabolic networks, gene regulatory networks, signaling networks, and by the end of the day, it's in these networks. Yeah, and I will show you examples of how this could look like in the following. Then over the last, now roughly 20 years, 15 years, there were two directions in systems biology. The first one was this omics idea, yeah? So whole genome sequencing, whole proteome, whole metabolome, you name it, they omic it, yeah? This was a direction that was mainly put forward in the US. And what they are doing still is that it is, on the one side, it is large scale, but it's still rather qualitative and still rather static. In Europe, it was more the direction that people looked at dynamic behavior. They used mathematical modeling to understand these networks, but this mathematical modeling was not just in the blue sky, as it sometimes has been in the field of mathematical biology, but it was very close to data, and why I show you in a second. Yeah, this is the so-called bottom-up. This was a top-down approach, and it is still on small scale. So when we started, the first model has 4 differential equations. Now we can deal with something like 50 players in this field, but it's still small scale. But it is quantitative, and it captures the dynamics. And you already see that this is really complementary, and what will happen by the development of new technologies is that these both approaches will somehow converge, and so that we hopefully finally come up with large-scale quantitative and dynamic models. But what both of these approaches are following is this idea that we have to look at networks. Yeah? Not single genes, not single proteins, but look at networks. Yeah, why do we have to do it in a database way? And this I will show you for one example. We already in the second talk on Monday saw this MAP kinase pathway. So here is this ERK, this MAP kinase stuff here. And this is a typical pathway cartoon. Yeah, structurally they all look the same. So you here I have a receptor at the cell membrane, and then there is some cascade. Don't worry about the fancy names. These are just names. And then down here there is then something that goes to the nucleus and initiates transcription. So this is a MAP kinase pathway. This is one of the most investigated pathways in cell biology. And now what you can see here is that we have here a positive feedback. These arrows means activation. And here we have a negative feedback. These little bars means inhibition. And now you can easily imagine, depending on the time scale and the intensity, the strength of these two feedbacks, they can show pretty different behavior. So assume that the positive feedback is much stronger and faster than the negative one. So forget about the negative one. Then if you have a transient input here, this positive feedback will translate it to a sustained output. On the other side, if the negative feedback is much stronger than the positive one, then a sustained input by the negative feedback will be translated into a transient, a sustained input will be translated into a transient output. Yeah, that means that you can have, this is depicted here, quite different systems behavior depending on the input and on the relative intensity of these two feedbacks. Yeah, and for example, this molecule RAS here is a very, very famous oncogene in many tumors. This is constitutively active, and so therefore changes this pathway. That means if we want to model this, we don't know all these parameters. Yeah, all these reactions are enzyme catalyzed, and you can't measure them in vitro. And so the idea here is we really have to take data from the dynamics of the system and then fit the parameters in this system. Yeah, so therefore it is data database. Yeah, and this is not only conceptual, but this is also happening in real world. So these are data from our most important Croatian partner at the Cancer Research Center in Heidelberg. So these are data from a cell line. This is a hepatoma cell line, so liver cell line, and these are primary cells from real mice. And what you can see here is the phosphorylation of a certain protein that is involved in the so-called TGF-beta SMAD signaling pathway. And what you can see here in the cell line for 2 replicates is that this shows a very transient behavior. Yeah? So it goes up and it goes down, while in the primary cells it goes up and it stays up. Yeah? This is exactly what this MAP kinase pathway, or what I showed you for the example of the MAP kinase pathway, and similar things happen in this pathway. And this makes biologically absolutely sense, because this pathway here with these SMADs molecules, the job of this pathway is to stop proliferation. Yeah, so you all might know that the liver can regenerate very well, so you can cut off half of it and it will regenerate to full size and functionality. And by the end of this regeneration, these pathways activated. Yeah? And in the cell line, it's a cancer cell line, yeah, their proliferation never ends. So therefore, things are changed in a way that this pathway cannot be activated in a sustained manner, while in the healthy primary mouse hepatocytes, this is sustainedly activated and therefore proliferation stops. Yeah? So it makes sense. That means if you have a certain system, a certain cell line whatsoever, you really have to take data from this specific system. Yeah? And what does not work out is just go to some database and from whatever experiment take some parameters for this model. This is really artifact research. Yeah? There were screens for parameters for different cell systems, parameters in these systems, and they differ by orders of magnitude. Yeah? So you have to look at the specific system if you want to understand a specific system, and especially if you want to understand, for example, the difference between a cancer cell and a healthy cell. Yeah. So therefore, what you have to do is you really have to do parameter estimation. And we are mainly dealing with deterministic dynamics that can be discussed in a lot of detail. And one reason is that in deterministic systems we understand very, very well what has to be done. Yeah? Of course, there is always stochasticity in this biological system. It's not temperature zero, yeah? Of course, there are fluctuations. But since we are mainly dealing with transient behavior, this transient somehow stochastic behavior can typically very, very well approximated by a deterministic system. Yeah? If you do, for example, Gillespie algorithm simulations, you see that for rather small numbers of molecules in a cell, you already get a behavior that looks pretty smooth. Yeah? This changes completely if you're working with oscillatory data. So if you have oscillatory data and you have stochastic in the dynamics, then it will not be possible, and this can be shown mathematically, it will not be possible to find a deterministic approximation for this. Yeah? And in the next talk, I think we will see some thoughts in this direction. Also, we do not deal with the spatial aspects. Lynn, in her talk on Monday, discussed different levels of abstraction for dealing with spatial effects. So we take here the middle level, so we take compartments like the nucleus, like the cytoplasm, but we don't really deal with PDEs. The idea behind is that diffusion, look at this poster, that diffusion in the cytoplasm happens on a time scale that is much, much faster than the time scale that we are looking here. Time scale here is typically something like 15 minutes, an hour, 2 hours, something like this. And within this time scale, diffusion plays no role. An important bit in detail is that typically one cannot observe these states directly. And typically one cannot observe all of these states. That means that the dynamics here, P are parameters, of course, and U with some external input like a stimulus here. Yeah, so that means that the observation is given by some function G. Please remember this G, it will turn up again. A mapping from the world of the dynamics to the world of the observations. And the dimension of this vector is typically smaller than this. The rule of thumb is you should have half of the players observed. Another thing is that typically measurement methods that sometimes measurement methods like Western blots and all these fancy biochemical stuff, they, for example, cannot discriminate whether a protein is phosphorylated, that means activated, or not. That means that in this observation function, we sometimes have linear combinations of the different players. Yeah, what also can happen is that you have a measurement technique that goes into saturation. There, that means that you here also might have some nonlinearity. Yeah, but typically we are here in the linear regime of the measurement methods, so that means that this G picks out certain components or picks out certain linear combinations. Yeah. Typically, these networks are sparse, so not everybody is talking to everybody. Yeah, you see for the MAP kinase, there's really a lot of structure in it. Typically, we have different time scales, so we have stiff systems, and they are externally different. Yeah, so what we have to deal with, therefore, is parameter estimation in nonlinear, partially observed, noisy. We have noise. 10% in the measurements. They are driven from the outside by some stimulation. They are stiff and they are sparse. Yeah? And we assume that here we have an error that is normally distributed. For a lot of measurement techniques, it's log normal distributed. So therefore, by taking log of the data, we come back to Gaussian noise here. Yeah, and then one can write down log likelihood. Yeah, sorry, I forgot an introduction. The first lesson we learned when we start dealing with real-world data is that if you deal with real-world data, by the end of the day, every question you have will lead to some statistical testing. Yeah, so we will see some statistics in the end, and parameter estimation is based on this log likelihood, which is a very general and powerful procedure. If you cannot come up with a log likelihood, then you also have to listen to Yannick's talk in the following. And if you want to see some extension to the spatial stuff, then you should attend David's talk on Friday, when he will talk about very cool mathematics to deal with this. But we are here in the nice situation that we can write down this log likelihood just by having the difference between the prediction of the model, the observed data, weighted by the errors, and then just take the square here. Yeah, it looks a little bit like regression, but of course here X is a solution of a dynamical system that makes it numerically a little bit more involved. Yeah, so now you have this chi-square, you have to minimize it, and therefore, and there are some technical things about this that I want to discuss after this more general slide. So George Box, one of the godfathers of modeling, once said that all models are wrong, but some are useful. And this is really a very, very deep sense, because you have to think about what is a useful model. Yeah, so a useful model should first be a good model. And this is discussed down here. So what is a good model? If your model is too simple, it cannot fit the data. Yeah, and in this process of modeling, typically, if you write down the first model and do fits, at least in our group, the first 5 models just cannot fit the data. Yeah, so what you're doing is you typically make the model more complex until you can explain the data. But then there are 2 other situations possible. Your model might be too large. Yeah, then you overfit the data, perhaps only slightly. But as a yeah. Yeah. No, you have to switch it on.
No.
You hear?
I'm sorry.
Yeah. Oh.
I just wanted to ask, so in this setting you consider basically data already existing, and you're talking about too large model, good model, or too simple model. Could you also argue that given data, a certain, let's say very little data, actually what was a too simple model could be now the model to select? Because if data does not have much information, maybe you should go for a...
Yeah. So just as a historical anecdote, when we started all this business, I came into contact with cell biologists, and I'm going to show you the data and the model in a few minutes. And then I said, oh, I need time-resolved quantitative data. Yeah, and she said, oh, no problem. And after a week, she came back and brought me time-resolved quantitative data. Yeah, and of course, there's a very simple model to explain this. Yeah, and then she went back to the lab and worked for 6 months on the measurement technique and produced the data I show you in a second. Yeah, of course, here flatliner is sufficient. Yeah, But this is an interesting point, and we come back to experimental design and model reduction towards the end of the talk. Yeah. So if my model gets too large, then I can perfectly fit the data, but the problem is that the parameters and thereby the prediction will not be well determined. Yeah, that means it's hard to falsify a model. Yeah, remember Newton's description of gravitation two-body problem. This was a very strong theory because it predicts that the planet is living on an ellipse. Yeah? And this, of course, is of measure 0 in the class of all possible motions, and therefore it was a very strong theory because you can test it experimentally, and therefore it was falsified in the end. Okay, yeah. So, and a good model, by the end of the day, it should be able to fit the data, but of course parameters and predictions should be well determined. Yeah. And so, if you have a good model, then it has a fair chance to become a useful model. And what should a useful model do? It should capture the main effects and neglect the rest. Yeah, and in neglecting the rest, for example, stochasticity, spatial behavior, whatsoever, in neglecting the rest, it is wrong. Yeah? It should make predictions that can be tested in the wet lab, and if it's really a good, cool, useful model, it should deliver insights that can only be obtained by modeling and by not whatever fancy biomedical technique. Yeah, and if you get this, then you have a fair chance for cell science. And nature. Yeah? So this is what we're trying to do, understand biology based on these models in a way that can't be done by whatever pipetting. Yeah? So, and this is this famous cycle, or these cycles in this business. So we start up with some prior knowledge. There has been a lot of work in figuring out how these networks qualitatively, statically look like. And then we construct this model. There are some first quantitative data. Then we calibrate the model, and then if we are able to, if we are then not able to explain the data, then we have to think about the model again. If yes, we have to do this uncertainty analysis, and then if it's well determined the predictions, then you should do really a validation experiment, meaning an experiment produce data that has not been entered the modeling process, and then if we can do this, then in the end we have the chance to get new biological insight. In the other way, we have to do this again and again. Yeah, the challenges here is we have to estimate these parameters. We have to look at these identifiability, the observability. We have to do experimental design, model reduction, and typically there is not one candidate, but there are a few candidates, so we have to think about model discrimination by the end of the day. So starting with parameter estimation. There are two general approaches to this. The first one are the so-called deterministic local optimizers. So the idea here is just you sit at a certain point and then you use gradient curvature information and in some intelligent way run down the hill, Levenberg, Marquardt, trust region, whatever. There are really a family of algorithms for this. But since you can only run down the hill, and typically the landscape of the square of the likelihood can have multiple optima, then the typical thing is that is done is you just take 100,000 of different random initial guesses and come up with the picture I show you in a second. The second class are the stochastic global optimas, and these guys are allowed also with a certain probability to run up the hill, and therefore they can just cross little barriers in the likelihood at landscape. And it is historically pretty funny that a lot of people say, oh, we have very, very many local optimas. You have to use this stochastic approaches. And this is, at least this is our opinion, this is not completely true, but relies on what is shown here and on the next slide. So if we look, and these global stochastic optimizers, they always apply a certain kind of heuristic search strategy, the genetic algorithm to do crossover mutations. Yeah, so. But at the end of the day, you have some magic factors that you have to fix here, and I come back to this in a minute. Yeah, so for these deterministic local optimizers, to find the derivative, you have to calculate the derivative of this observation function with respect to the parameters. This just turns out if you write it down. Yeah, and then just this looks like this. Yeah, and it's very easy to derive this one. it is very easy to derive this one, but it's difficult to derive this one. Yeah, and what people typically do is then they take finite differences. Yeah, so meaning they approximate this derivative here by this finite differences in different directions in parameter space. Yeah, and it has been shown in the early 80s that this does not work out, because to derive, to come up with this, you have to integrate the differential equation. And if you integrate the differential equation, you typically apply some step size control. And what can happen, and I'll show you an effect in a second, is that when you integrate it with a little bit different parameters, this integrator chooses a different step size control than the other one. Yeah? And then, of course, you have this approximation or the choice of these h here. And what has been shown is that these two kind of errors, so the introduction here by this first order approximation and here the different step size controls, that these two kind of errors cannot be controlled. Yeah? So anything can happen, and I'll show you what happens in the following. But there is a cool way out of this, because you can just solve the sensitivity equations. And what can be shown is when you integrate the system and you integrate simultaneously the sensitivity equation, that the numerical burden more or less does not increase. Yeah? So this is a very elegant way out of this dilemma, not being able to control finite differences. Yeah, and the effect is shown here. So here is some scan of some parameter, and here is a calculated derivative for different choices of these h. And what you can see, if you choose it very, very small, you get this very spiky behavior. And these are just those points where the two trajectories for the finite differences switch their step size control in whatever manner. Yeah? If you choose h a little bit smaller, you see it becomes less bumpy, and if a little bit larger. And if you choose this much larger, you have the green curve. This is now pretty flat, pretty smooth, but you see it is biased. Yeah? And if you do it by sensitivity equations, you get, with 10 times faster than finite difference, a very nice smooth curve. And now you can very easily imagine, if these are your calculated derivatives and you do optimization with this, yeah, it will completely destroy the likelihood landscape. Yeah? So therefore, if you do this, your chance is very, very high to get stuck somewhere where, in principle, the likelihood is really smooth, but by having these incorrect differences, you just produce holes all over the place. Yeah, yeah. So whatever your famous optimizer is, there's a very easy way to check out whether it's a good optimizer, and this is shown in principle here. So we have two parameters here. We have a likelihood landscape, and we have 4 local optima. This is the deepest one. This is the second one, third one, fourth one. So if you have a good optimizer, even a local one, the expectation is that it at least finds a local optima. Yeah? And this is shown here for these blue little curves. Yeah? You start somewhere, and then it goes to this local optima. You start here somewhere, it goes to this local optima. If you have a method that does not perform well, for example, because you calculate the derivative in the wrong way, then it will just get stuck somewhere. Yeah? And you can now transfer this plot into what we call the waterfall plot by just sorting the final hits by the likelihood and then plotting the likelihood or the chi-square by the end of the day. And for this blue good performing method, you see these 4 nice steps. That means we have reached an optimum. Yeah? And here for the not so well performing, you see that's the slope, and these are stuck somewhere, yeah, on the way, but you don't see these steps here. Yeah? And from having plots like this, then you can argue, yeah, this should be the global optima. Yeah? And typically we don't do it 50 times but 1,000 times, and then you get some confidence that this is the local optimum. Yeah, and we compared now global optimizers, stochastic ones, to the local deterministic ones, and took from a group in Tübingen. They assembled a dozen of these different algorithms here, and we did this plot, and you see there are no plateaus here. Yeah, the comparison is not completely fair because we just took the default values that they put in for these magic factors. Yeah, so if you have a lot of experience and tinker around, it might become better. And now if we compare now, so these are the curves from before. So this is finite differences with local optimizers, and these are the sensitivity equations. Yeah, and you see there are orders of magnitude between this. Yeah. Yeah. So the problem with these stochastic optimizers is that they at least contain whatever language of fancy genetic or biological or neural net or whatsoever you buy, they contain at least two magic factors. Yeah, and one of these typically corresponds to gradient and the other to curvature information. Yeah, meaning you have to know the solution of the true problem to choose this in an optimal way. Yeah, and therefore it is kind of problem. And what we did here is typically what is called the initial value approach. That means we start with some initial values for our trajectory, integrate it up, and then make this comparison. This is fine for transient processes, but for oscillatory or even chaotic processes, this does not work out. And this has a very, very vivid reason. So assume you have an oscillation with a certain frequency. Frequency is one of the parameters. And if this frequency in your initial guess is a factor 10 off, you get a solution like this. And you will never be able, by minimizing chi-squares, to stretch this wrong. Yeah, is it clear? Yeah, to stretch this wrong amplitude. And therefore, in the 80s, a colleague from Heidelberg has developed what's called the multiple shooting, and this is a very, very sophisticated. So he published 2 conference contributions about this and became full professor for these 2 contributions. So this was really a very, very nice idea. But you only need it if it's oscillatory, and since here we have typically transient one, then we are fine out with this much simpler initial value approach. Yeah, we deal with noisy data. That means in our chi-square we have this sigma squared, the variance, and typically what is done is that this variance is estimated in the very naive way by just calculating the mean, taking squared here, normalize it correctly, and then get an estimate of the sigma squared. The problem is that typically biologists measure 3 replicates. And now it's known from statistics that this estimator here is distributed as chi-square with k minus 1, in this case, degrees of freedom. And if you have 3 replicates, you have the chi-square distribution with 2 degrees of freedom, and this is just the exponential. Yeah, that means for the exponential, you get a lot of very, very small values, more than you expected if it would have been a Gaussian. But since this decay is only exponential and not exponentially squared, you also get much more than expected of large values. Yeah, and this can hamper the estimation in a drastic manner. So therefore what we do is that we estimate also this chi-square based on some error model. And this error model typically depends on what we have observed, what is predicted by the model, and there are some parameters. Yeah, and typically we take something like an absolute and a relative error, and therefore we don't have chi-square anymore, but now we really have a likelihood, because here are also parameters in this term of the error. And doing a little simulation study by just looking at the process where A goes to B and goes backwards. So we have as parameters initial value of A, initial value of B, and these 2 rate constants. And here we have the comparison in red if you estimate it from 3 replicates, and in blue if you estimate it based on an error model. And here we have the log ratio. Yeah, so 0 means it's very good. And you see that the variance is much smaller if you base it on an error model. Yeah, and so these are little tricks to take as much information from the data as possible. Yeah, now coming to quantification of parameter uncertainty. And I will start a little bit with the classical, then introduce this profile likelihood. This is the main contribution of our group in this field, and compare it a little bit to the Bayesian approach. There are 2 types of non-identifiabilities. The first one is a structural non-identifiability. And the setting here is, again, you have this dynamics, and you have a certain observation function, and we assume that there is no noise, so best of all worlds. And the question now, if we know the input, if we know the functional form of f and g, and if we have data, can we determine p in a unique manner? This is a question of structural non-identifiability. And if we have such a structural non-identifiability, that means that we get a relation between the non-identified parameters. And if we walk on this manifold of non-identifiable parameters, then the prediction of the model, and therefore the likelihood, does not change. Yeah, assume the very simple setting, regression, y is a times b times x. Of course, you can never determine a and b, but only a times b, meaning if you double b by a factor of 2, you can just reduce a by a factor of 2, and you have the same prediction of the model.
Are you assuming that your data data is no noise in it.
In this case, we take the best of all worlds, and we assume that the data is noise free. We come to the noisy case in a second. Yeah, because here we are only talking about the structure. Yeah, and if you have such a structural identifiability, then there are two things to be done. Either you can change G, meaning you measure additional proteins, or you have to reduce somehow this function here, for example, by reparametrization. In this case, if y is a times b times x, you can just replace a times b by c, and then you get an identifiable one. Yeah? So the second thing is the practical non-identifiability. And experience shows that this is a much more important case. This has not really clearly been defined in the literature. There were statements like, the errors are large, yeah, but this is somehow right. Yeah? And this is due to poor data quality. Should never say poor data quality to biologists. Yeah? But the point here is that if you are in this setting, then if you change the parameters, then the prediction of the model and the likelihood, they change slightly. And what slightly means, I show you in the following. Yeah, and the way to deal with this classically is based on the Fisher information. Who of you knows what the Fisher information is? Who of you does not know what the Fisher information is? Rostock, no. So, the idea here is the following. If you have your estimated parameter, p hat here, then locally the chi-square function will be a parabola. Yeah? Globally it can look whatever, but locally it will be a parabola. So the idea here is, if you change this parameter and the chi-square only changes slightly, then the parameter is obviously not very well determined. If this curve looks like this, the parameter will be much more determined, yeah? Because if you change p, you will see it in your chi-square function. Yeah? So, and now assume you are in the asymptotic case that this parabola behavior, the range where it behaves like a parabola is large enough, then you can show, and this is a lot of calculation, that here we have chi-square, that there is a certain threshold, and this can be calculated from the chi-square distribution. Yeah? There's a certain threshold, and if you take this values here, then you get confidence intervals just at the intersects of the increase of the chi-square of the likelihood. Can one still see anything here? Yeah, I will come back to this in a second. Yeah, so that means I take the curvature here, because the curvature, yeah, determines whether it looks like this or this. If I take the curvature here, I can calculate where these cross sections here with this critical value is, and then I can come up here with confidence intervals. Yeah, this is the classical Fisher information theory, 100 years old. Yeah, but it assumes that I can extrapolate from this point only by the curvature, yeah, to the outward. And we will see in a second cases where this is not the case. Yeah, so this is the good old idea. Here you take the curvature at the point of your estimate, and then you get this asymptotic confidence intervals just from the inverse here. Yeah, if curvature is large, confidence intervals are small, and the other way around. This is perfect for linear models. Yeah, for linear models, this parabola holds globally. But keep in mind that the linear stochastic differential equation in the solution space is a nonlinear model because it's an exponential. Yeah? Only the differential equation x dot equals constant produce a linear solution. Yeah? Meaning for all cases that are non-trivial, we are in the nonlinear regime, so it depends how good this approximation is. Yeah? And the problem is if I reparameterize the system, and typically we live in log space concerning the parameters because this is somehow the natural choice, then this is not invariant under reparameterization. Yeah, so therefore, what we are using is a so-called profile likelihood. This is also an old concept from regression theory in the 70s, in the 80s, and until today I don't understand why it's not used anymore, because it's really a very cool thing, like I want to convince you in the following. So the profile log likelihood, I typically skip the log, is the following. It is defined per parameter, and you minimize the likelihood again by keeping this parameter fixed and minimize over all the other parameters. Yeah? In two-dimensional space means you scan here from east to west and re-optimize north-south. Yeah? And then you take the other parameter, you scan north-south and re-optimize in east-west. Yeah, so you take the likelihood with all other parameters re-optimized for a fixed, yeah, you scan this one for a fixed one of this. Then again, statistic tells us that we can derive at confidence intervals. So if we take this minimal value, if we scan the parameter, typically it will increase, yeah, and this then should stay below something that's given by the chi-square distribution. This is just some critical value. It's 3.5 for one parameter or something like this. Yeah. The cool thing here, it is reparameterization invariant, and it is much better properties compared to the Fisher information. There is an asymptotics, but it's a very, very weak asymptotics. Yeah, so meaning you reach it very fast, and we will see examples for this in the following. Yeah, so just to give you an example, what it means. In the green case here, yeah, we have one parameter. Here is the optimum. Here is chi-square. If we have the green case, then we are perfectly identifiable, and here the result of these classical Fisher information will be the same as for the profile likelihood. The other extreme is that this is a completely flat curve. Then we have this structural non-identifiability. And then what we can have is this practical non-identifiability, meaning we have one direction where we don't cross this critical value. Yeah, that means this one here is statistically consistent with being 0. Yeah, and here we can perfectly introduce confidence intervals. Here we only have a lower bound, but we are completely open to the other bound. Yeah, and if you apply the asymptotics for this blue case, and we will see examples for this in the following, of course you always get finite confidence intervals. Yeah, where the confidence intervals include infinity. Yeah, so this is wrong in the wrong direction. If we have practical identifiability, we measure more data, then this might move up. If we have this structural, we have to think about the structure of the equations, model reduction, augmentation of the observation state. Yeah. So the next concept is the non-observability that is very closely related to the non-identifiability. Again, we are in the best of all worlds, noise-free observations. And now the question is, if we know input, if we know all the structure, if we have the data, and is the state X uniquely determined? Yeah, remember, we don't observe the state X directly. Some components are not observed at all. And now you can easily imagine if some parameters are not identifiable, then some of these states will also not be identifiable. Most often this non-identifiability is introduced because we only have relative measurements, fold changes. Yeah, you can easily imagine if you have a fold change parameter and this is then translated into the dynamical system, and then there is a fold change parameter times the kinetic parameter, yeah, then you get this kind of exchange between these two parameters that then lead to this non-identifiability. Yeah, to give you an example. So this is one of the simplest pathway, and this is a pathway we started with many years ago. It's called the YAK-STAT pathway. YAK stands for Janus kinase because it has double-headed stuff, and STAT is an abbreviation for this molecule, signal transducer and activator of transcription. And what you can see here is a scheme of it. So here is a receptor. STAT gets phosphorylated. It forms a dimer. It goes to the nucleus, and it can be exported as a monomer dephosphorylated again. Yeah. This is the input, the activation of the receptor. These are the data for the total amount of cytoplasm in the cytosol, and this is a phosphorylated one. Yeah? And you see here this function G, so this is the total amount, so it is the unphosphorylated monomer, the phosphorylated monomer, and the dimer. These three are combined, and this then is a monomer phosphorylated and the dimer phosphorylated. Yeah, so we have this typical kind of nonlinear combination, and we have two observations that enter the estimation, but we have 4 players, so that is this rule of thumb of 2 that you should have. Yeah. Now, if you estimate the parameters, so we have here these scaling parameters for the observations, we have the initial guesses, and we have the dynamical parameters. These are the profiles. Yeah, and for the first one, this looks perfect. Yeah. The second one is obviously structural unidentifiable. This looks rather wiggly. You see here the asymptotics from the Fisher information, and you see that this goes completely wrong because it tells you have a finite confidence interval here, while the profile tells you that this stays flat here. Yeah. Here again, this gives you finite, but the truth is much larger. These little steps in here come from the fact that if you scan parameter space and you have multiple minima, yeah, then you can just jump over the cliff and go to the next minimum, and this is exactly what happens as these sharp steps. Yeah, and here now is again non-identifiable parameters. So if you now look at the observability, and we look at those parameters who are structural non-identifiability. Yeah, there were 4 were coupled, but on a one-dimensional manifold, as the analysis shows. So what happens if we go along this manifold, then the prediction of the model will not change, but the internal components, and they scale. Yeah, so if we move along this manifold, the states of the system, they change. Yeah. And what you can learn from this plot now is experimental design. Yeah? So for example, it would not make much sense to tell the biologist, measure this guy here. Yeah? But it would make a lot of sense, measure the initial condition, or measure something around here. Yeah? So you can turn this around and do experimental design. The same thing you can do for this practical non-identifiability. Yeah? And here I also plotted the prediction of the model, because this changes slightly, and here you see exactly what slightly means. Slightly means the trajectories change in a range that is consistent with the error bars of the data. Yeah? This is slightly changed, but it's statistically not significant. And what you see here is that there are 2 internal components that are not affected by this, but 2 other components where this one goes up, this one goes down, they are severely affected. Yeah? So from this plot, you learn it's not a good idea to tell the biologist measure this or measure this to get the model identifiable, but measure either this or either this around 20 minutes. Yeah? So that means there are 2 more data points that are necessary to fix the problem. Yeah? And the cool thing is you can now first simulate what happens. Yeah? So for example, assume that the measurement of this component at t equals 0 gives something like 200 plus 10% in whatever units. Yeah? And this one gives this in whatever units. Yeah? And then you take these 2 additional data points and do the whole analysis again. And this was before and this was after. Yeah? That means more or less all problems, yeah, here's a little bit left, yeah, all problems are solved, and the gray curve and the black curve is more or less the same, so means we are in this asymptotic regime. Yeah? So that means you can really do a price list. Yeah? If the biologists want to have the model very predictable, we can tell them the price you have to pay is give us these 2 measurements. Yeah? And you can play around with this, and then in the next round you can also fix this guy. Yeah? So therefore, based on this profile likelihood, it's very easy to make experimental design to get models identifiable. And the cool thing about this profile likelihood is because it's, yeah?
If the 2 wildly varying parameters compensate each other, like entropy and enthalpy to give free energy.
Sorry?
Well, you showed basically some measure which is varying very slightly, and you found 2 other measures which both very big. But what if they compensate each other? And think in terms of
if they compensate each other, the result would not look like this. Yeah, this says everything is, apart from this little bit, yeah, this says everything is well determined. Yeah, if something could be interchanged in the model without having an effect.
Oh, no. I'm thinking, let's say entropy and enthalpy.
Entropy?
Entropy and enthalpy. And you're actually measuring free energy. Like, you're interested in free energy. You measure huge variation on the entropy, you measure huge variation on the enthalpy, but the actual determining force is the sum of those 2, so it's not going to help the model, does it?
No, but this would, in terms of this idea, this would then be a structural non-identifiability. Yeah? when you can compensate the effect of one change by an effect of the other change.
They actually kind of coupled in the result.
So this will then result in these flat curves.
Something like that. Exactly.
And that tells you have to think about the structure of your model. Yeah, this is the same as in this simple case. If y is a times b times x, then you can never identify a and b alone. You can only get the program.
Right, but it takes huge effort experimentally to measure. So both components of a flat curve are actually difficult to measure.
Exactly, we cannot measure the parameters. Therefore, in case of the structural identifiability, we have to think of an extension of the model to make either the model simpler or to increase this observation vector, and this is just done by these two guys. So these two components did not enter the model estimation, parameter estimation in this case. We here had only linear combinations of different players. So we changed this important vector G, which couples dynamics to observations, and if we increase the size of this, we have the chance to learn more about the system. Does that answer your question? Okay, thanks. So, since this is calculated per single parameter, it scales in a linear way. You can do it on parallel computing, and this is just one, and we do this calculation in the group every day. So it's really a very versatile thing. And this is just one example. I have completely forgotten what it's about. So there is some before and some after. So something in the model is changed, and you can see that there are certain cases where this has exactly no effect, and other cases where it has a large effect. And there are also cases here where we have, for example, only an upper bound for the confidence and nothing for the small values. And I will come back to this when we talk about model reduction. And the method has the following properties. We don't need any assumption on how this manifold of non-identifiable parameter look like. You can apply it to large system because it's per parameter. And you can apply it to any parameter estimation problem that is continuous, as long as you have a likelihood. But some of the ideas will also take over to these more general methods we will hear in the following. I showed you how to do experimental design. Will come to model reduction, and the general goal here is that we want to have models where the complexity of the model, all models are wrong, some are useful, where the complexity of the model is really tailored to the information content of the data to be sure that we have good predictions. And the most important thing is that this idea is very easy to communicate to biologists. I will come back to some Lie group theory-based methods in the following. This is something you cannot communicate to biologists, but this is something, because it's so easy graphical, that it's very easy to show to biologists. There is, of course, the Bayesian idea for uncertainty analysis. That means essentially Monte Carlo, Markov chain Monte Carlo sampling of parameter space, and we did some comparison between these two cases, and of course the sampling is very, very expensive numerically, and if you have complex geometries in parameter space, 50 dimensions, that is very hard to control. And typically, you have to choose some kind of prior. That might always be an issue. And it is especially problematic in the case of these structural non-identifiabilities, because in the structural non-identifiabilities, you have manifolds you can move on and nothing changes. And if you sample this in a Bayesian sense, you can sample until the end of all days. And the problem especially is if you have chosen some non-trivial prior, then this non-trivial prior on the parameters, this will hide all structural non-identifiabilities because you just then sample the prior. The likelihood is flat in this case, but the prior introduces structure, so you will not be able to see structural non-identifiabilities. And bottom line, if you really have a good model, everything is fine. Then the results of the Bayesian approach and the profile likelihood is just the same, but the profile likelihood typically is much faster. Very, very short. For the structural identifiability, there are analytic approaches, and the idea here is in principle very easy. So if you have a manifold in parameter space that you can walk on without a change for the prediction of the model, that means that you have some symmetry. And we know from fundamental physics that Lie groups are very good to deal with this. Yeah, so that means you have a transformation that transforms your state vector, your parameters, and your input in something else in a way that this prediction of the model does not change. Yeah, and if you have such a transformation, then you can discover it and take use of it. And there are, in this theory, there are the so-called infinitesimal generators, and the simplest one, this x in the theory, that corresponds for our systems just to scaling of the variable. Yeah, and as I mentioned before, this is one of the most often cases for introducing structural identifiability. Yeah, and then there is a really magic thing. Yeah, the next complex, x squared infinitesimal, that corresponds to Michaelis-Menten saturation. Yeah, which might hold instead of mass action for the dynamics, or in the observation form, in the observation function for saturating measurements. Yeah, and this is really magic, because this two theory, identifiability analysis, dynamical systems on the one side, and Lie group theory, they are completely independent. Yeah, but taking advantage of these, we have implemented a very, very fast method to identify the structural identifiabilities, and they can typically just fixed by fixing parameters. Yeah, so this is the easy thing. The practical identifiabilities are a little bit more complicated. Yeah, model reduction. What typically happens is you start with a model that cannot explain the data. You make it larger and larger and larger. Then you can explain the data, but what typically happens on the way is that you introduce some complexity that is not necessary to describe the given data. So that means you end up with a setting that your model can describe the data, but it's too large, and therefore you get in all these identifiability problems. Yeah? So that means you have to reduce it again. This is, as I mentioned, easy for the structural ones. You can just reparameterize the model. But for the practical identifiability, it's a little bit more tricky. And it's, again, the profile likelihood that helps you here. And because there are 4 cases that can generically happen. Your profile likelihood for a certain parameter may stay below this threshold here for and or either plus or minus infinity. Yeah, we are on log scale, so therefore plus minus infinity. And then what can happen is that the remaining parameters, they change when we scan this, or they do not change if we scan with this. Yeah, and this means 2 possibilities here, 2 possibilities here. That means we have 4 cases, and for all these cases there is a specific cure. I will show you this for lumping states. So let me see whether I get this. So if it stays below the threshold for plus infinity and it's not coupled, then you have to lump states. If it's coupled, you have to lump states. If it's not coupled, you can remove reactions. And then for this case here, you find situations where you can just replace an ODE by an algebraic equation because it's so fast. And in the other case, you can just fix variables. So that means we can deal with all of these possibilities. And I will show it for the first case. So we have a reaction here where X gets phosphorylated, gets double phosphorylated, and we observe only the double phosphorylated state here. We want to estimate these 2 rate constants, and if we do this in a naive way, we get this profile likelihood here, and the first thing you see is we have 2 optima here, and this means just that we can exchange these 2 parameters with each other. Yeah? So this is just a point transformation, but the important point is that this one stays below the threshold for large values here. Here. Now if we plot the dependence, when we scan this parameter and we plot the dependence of the other parameters on this, we see that for large values, they are not dependent. Yeah? So this parameter does not depend on this one. Yeah? And therefore, it says we can lump these 2 states here, just delete the one in the middle and get one reaction, and then everything is fine. Yeah? And what you can see here, these are simulations. Yeah? These are simulated data points, and you can see here before and after the reduction, and you see no difference. That means this is statistically fine. We can still describe the data very well, but now have all parameters identifiable. And this can be done also for the 3 other cases. I mean, we have a more or less automatic procedure of how to get back from a little bit too large model again to a good model. Yeah, you can also apply this idea to the prediction uncertainty. So before we had the parameter profile likelihood, now we have the prediction profile likelihood. So that means we have whatever prediction of the model, yeah, everything that can be computed from the model. It can be the ratio of the maximal value of component 1 to component 7. It can be whatever, yeah, the area under the curve of something or whatever. And you can exactly apply this idea of the profile likelihood also now to predictions, yeah. So you have some function of the parameters that should be a certain value set. You screen for this prediction set, you minimize over the whole stuff again, and then you get these confidence bands by the same procedure as before for your prediction, yeah. It can be anything that can be predicted by the model, and if what is predicted is one of the states that are not observed experimentally, then by the same line of argument as before, we can define these non-observability, the practical and the structural one. Yeah? And the cool thing here again, it's a one-dimensional problem that you have to solve here, and you don't have, like in the Bayesian case, sample high parameter spaces. Yeah, and just to give you a little impression, a little bit complicated. So here we have time, yeah. Here we have concentration, and here we have this value of these prediction profile likelihood, yeah. And whatever it is, yeah. So we have two setups, setup A and B, and you can see that for the black one, setup B, that here you have this nice parabola, yeah. That means for this state that is observed here, certain hormone stuff, yeah, the predictions are very well determined. If we change the other setting, B, you see that this one gets flat here, yeah? So it's not determined to smaller values of this concentration. Yeah, and this can be helpful for two things. So if you want to improve your parameter estimate, yeah, then choose an observable that is a variable that is non-observable, yeah? So one where this is as flat as possible, because if you then measure it, it will be fixed, yeah? So it's a very informative measurement. Yeah. But if you want to select models, yeah, then you choose very well determined, very well observable variables, yeah, because if you have this prediction, small prediction here everywhere, then it's very easy to falsify a model. Yeah, so therefore this helps with respect of parameter estimation and with respect to model selection. Yeah, and with all these methods at hand, we can now really go the full circle. So we can start with some data, talk about parameter uncertainty in parameter space, then talk about these prediction uncertainty, do model reduction, to model augmentation, and can then go back to predict something new in the data space to validate the model and hopefully then use the model to get new insights. Yeah, to summarize. So we are dealing with optimization problems since we want to fit it to the specific system under investigation. There are a lot of different optimizers out there, and whatever your famous one is, watch out for these steps in these waterfall plots if you see these plateaus. If you see the plateaus, it's absolutely fine. If not, you are in trouble. These parameter profile likelihood that turned out to be very, very, very informative. First, from the mathematical point of view, we get these finite size confidence intervals where we have cases where these asymptotics here does absolutely not apply. And keep in mind, if the true curve looks like this and telling you there is no upper confidence interval and you do the parabolic approximation, then you really lie to yourself, because yeah, you then claim that confidence intervals are much smaller than they really are. Yeah, and this is highest penalty. Yeah, we use it a lot for this model reduction in model building, and then on the other side, this prediction profile likelihood can very well be used for experimental design and for model discrimination. Yeah, I don't know whether you are familiar with this because we are so different here. No, this we can skip. Yeah, perhaps since in one of the talks we heard one model to rule them all, I will spend 1 minute on this. So often the question is, you have a healthy cell and you have a diseased cell. Cancer is a good example. Yeah. Then these two cells are not completely different, yeah, but something is different. Yeah, typically you have mutations. Mutation leads to overexpression of some protein, overexpression of some kinase, overexpression of whatever. Transporters in the nucleus are changed or whatever. Yeah, but the typical idea is you have a few of these mutations. That means in mathematical terms, since all the quantities in the model correspond by the end to proteins, yeah, initial concentrations are proteins, enzymes are proteins, yeah. That means what we try to do is we want to have one model to rule them all, yeah, meaning we want to have a model that describes the two cell types, but that tells us that at minimal cases where these models have to be different. Yeah, so that means what we are doing, we're doing a kind of regularization step, and we do L1 regularization if you are familiar with this, because this leads to sparse solutions, and penalize the case that parameters are different. Yeah, so that means we start with a very high or very loose, that does not matter. Yeah, but let's say very high, yeah, penalization strength, so that the penalization forces the two models to be identical. Yeah, but then of course it typically cannot describe the data. Remember these data I showed you in the very beginning, and then we relax it always a little bit more until we can describe both data sets with a minimal amount of difference in the parameters. And this is again done by the profile likelihood. Yeah? So that means we can identify cell type specific parameters, and we did it here for some healthy and for some lung cancer cell. And the cool thing is then, if you then do sensitivity analysis where you can best interfere with the pathway, because it's nonlinear, typically the best points of intervention are not identical to the points where the systems are different. Yeah, and this is again a case that can only be revealed by a model. Yeah, you cannot do this experimentally. It's much too complex. Yeah, so this is again something where we augment the techniques of biology by this mathematical modeling, and then and this is model-based functional genomics. You can of course then look up whether those points where you see the difference are also genetically or epigenetically different. Yeah, so you can really come back to biology here. Yeah, and therefore I would like to thank the people who were involved. So Andreas Raue has more or less initiated all this, and these and also many different people contributed to this. And all this has only become possible because we now have a 20 years collaboration with the group of Ursula Klingmüller and her co-workers who are now on the at the German Cancer Research Center in Heidelberg. Yeah, and if you want to have a short look on this stuff, so most of it is summarized in these papers concerning the optimization, and then review of what profile likelihood in systems biology can be done is given in this paper. And everything has been coded in a software. It's called Data 2 Dynamics, D2D. It is open source, so it uses MATLAB, so you have to have MATLAB, but otherwise it's open source. It's still growing, and if you want to find it, the easiest way might be Google just Andreas Raue D2D, and then you can find it. And yeah, and I hope we had a little more of students here, but for everybody who looking for a new job, so if you are interested in developing methods and also applying it to real-world data closely together with, then send me email. And I thank you for your attention.
Thank you. So just a quick question comment. You mentioned in the beginning the importance of or the fact that adaptive step size in an ODE solver could potentially destroy the gradient, basically, or make it very noisy. I just curious. In today, most later development of ODE step size regulations are such that they never reject steps. So this makes them much more continuous in the tolerance parameter, for instance, and also more continuous in small parameter perturbations. Just of interest.
That might improve the situation, but the cool thing about the sensitivity equation is that this is a perfect solution to this problem. Yeah, so this is investment in the wrong, concerning this problem, this is investment in the wrong direction, because the solution is already there and much faster.
Sure. Okay, and then just a quick comment. So you mentioned Markov chain Monte Carlo, and one thing that you lose when you're looking for point estimate with uncertainty bars is that it's going to be difficult to a posteriori test hypothesis. So let's say you have data and you have your model and you do you find a posterior distribution of parameters. Yeah. Then you could actually sample those parameters, let's say 100 of them, and you can test your model with respect to any hypothesis that you might have of the real situation. When you have a point value and an uncertainty bar, you cannot really sample the situation a posteriori, and you do not really get a statistical proof, right?
No. So this is a problem that is solved perhaps a little bit too much, but this is so if we have a model prediction, we want to validate it. We don't look at parameters plus confidence intervals, but we look at this prediction profile likelihood, where we optimize in the whole parameter space. Yeah. So there we are. We have the full dimensionality again. Yeah. But if we have let's assume we have a scalar prediction. Yeah. Then we have a one-dimensional optimization problem for this prediction, but we do the optimization in the full parameter space. And so we produce a one-dimensional curve in the full parameter space with respect to this prediction. So we don't do it here based on the parameters and change this a little bit, yeah, but we do it in the full dimensionality. But there we do it in a one-dimensional way, and this makes it much faster than sampling high-dimensional parameter spaces.
So maybe my question was unclear. So just the point was that you can test an hypothesis a posteriori. You have a posterior distribution and you have a question, what does your data support? Yeah. And it's not clear that you can do that by this method. Here you have
could I have the slides again? So the point here is, so this is a prediction profile likelihood, meaning I have a certain prediction, ratio between two values at time point 10. Yeah? So then I screen this prediction, I minimize, and this is the whole dimensionality, I minimize my chi-square, my log likelihood, with the constraint that the prediction of the model is this value. Yeah? So while in the Bayesian setting, yeah, you sample all over the place, yeah, and what we're doing here is we find a one-dimensional curve, yeah, through the whole parameter space, yeah, through the whole parameter space, that gives me this prediction. Yeah, this reduces the complexity, but we are not at the single parameter plus minus confidence. We are in the full high-dimensional parameter space. Yeah, and this is numerically very, very efficient.
Thanks, Jens, for this great talk. Yeah, so I know the D2D software, so just a question. So this is updated all the time, so all this what you presented is now already included in this?
And much more, yeah.
In this. And are there any, for example, this works only for ODEs?
Yeah.
Are there any things to maybe extend this to some spatial stuff or some any ideas to?
It is open source on this GitHub stuff. Whoever wants to contribute is highly welcome.
Or
and there are other groups than our who contribute to this.
Do you have any ideas or any? Is there any plans to introduce spatial components into this framework?
So far not. So far not. Our attitude is typically that we only look at new methods if the projects calls for them. Yeah, but of course, everybody can contribute. So there are different groups. Also, people from Berlin have contributed, and highly welcome.
But how do you? I mean, I work with this. Sometimes I had some. I modified by myself on my own version of this, but I didn't upload it to GitHub because
that's a pity.
Yeah, but who is now in charge of taking care? Whether it can be sometimes it can have also deteriorating maybe consequences. So who is validating all of this, the changes, if many of them contribute?
Luckily not that many, but send an email to Andreas Raue or send an email to me, then I distribute it to the people who do the jobs.
But Andreas now, I think, is in a company, works in a company? Or
Yeah, but he's still working on this.
Mm-hmm.
Yeah, it's his child.
Any further comments? I had one. I think PDs, you could potentially deal with PDs. I think it's no problem at all to.
Conceptually, it's not a problem. No.
This is, I don't think it's, as long as in your G function, which is the operator, you take solutions from the PD.
Yeah, no, conceptually, it's no problem.
Yes.
Yeah.
So we have this actual approach.
Yeah, and PDEs are, in principle, is at least my fingertip feeling, are in principle easier than stochastic differential equation because you still have this likelihood.
But you have the price of solving the PDEs.
Yeah, but this is all of you know how the yeah. Yeah. No, we have also done a few projects that involve PDEs, but for the daily business in the signaling, our feeling is that we don't need them. Yeah.
So in practice, you use this for estimating what's the size of the parameter space where you have used this? Is it like 5 parameters, 7 parameters?
As I mentioned, we are dealing now with systems 50 dimensions of state variables. That means we have 50, 60, 80 dynamical parameters. What we very often have is that the biologists do experiments under different conditions. Yeah, so for example, you do a knockdown, you do an overexpression or whatsoever. And these are experiments that are very, very informative for parameter estimation. And since typically experiments are expensive, and typically they measure then on relative scales, we have a lot of these observation functions where we have a lot of these scaling and offset parameters in. And these can be very, very, very hundreds, a lot of them. Yeah, but from the dynamical systems, we are now in the scale of 50 players. Yeah, so we started with 4, are now at 50. That means in 10 years we are now, let's see. Yeah. And the rule of thumb is the complexity of the models, the size of the models is not an intrinsic problem as long as you have enough experimental data. Yeah? And the rule of thumb is you should have at least half of the number of observations should be half of the number of players. This is my personal rule of thumb. Yeah. We had examples with high precision laser experiments where we had 10 equations and 1 observable was there. Yeah, but the data were high precision and everything was fine. Yeah, absolute measurements, no errors, no noise. Yeah, but in biological data where you typically have error bars like this, rule of thumb is half of players should be measured.
I understand. But because I thought, one of the key limiting is usually to have the correct underlying models, because many times, including, what we had with Jürgen when we were looking at small system and already with 5 equation, or this is already a headache because actually you can go with this model and actually the model is not working because we're missing a player, and then everything becomes incorrect.
Yeah. Sure.
So is there? I mean, sometimes just to finish the question. So, how do you? Is there a way to say that finally you are not satisfied? You are not satisfied with what you're obtaining here, not in terms of minimization, but you are not satisfied, and then you have, in some sense, to reject what you have. And this is, I think, a feedback to the community to have a sort of tool to say, look, the model is not accurate enough, something is missing. And this is the feedback, actually, that we need.
Yeah, this is a very typical experience, of course. Yeah, and in the very, very first project, where we first got this data here, after we got the data that I showed you, that was the first project, the first model they gave us was wrong, because there was one step was missing in the model. Yeah, and the first fit we had looked like this. So the data, looked like this for one component, and it looked like this for the other component. Yeah, and the best fit of our first model looked like this. So we were able to describe this data more or less, but for the red data, the best fit of our model was this. Yeah, so it was not necessary to be a world-leading statistician. It was just plain wrong. Yeah, and the point was there were one reaction missing in the scheme they gave us. Yeah, and because it was so simple, we could figure out there were only 3 possibilities what was meaningful to change. And we changed this one thing, and then we can perfectly describe the data, made predictions that were tested experimentally, blah, 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 yeah? Then everything was fine. But there is, so, because you have to have intuition and experience, my personal opinion is that computers will never take over the modeling stuff, yeah? Because you really have to think about steps when it does not work. Yeah. But the nice thing with the signaling pathways is that there is a lot of qualitative knowledge out there. Yeah. But typically the first models do not work. Yeah.
Last question.
Yes, I'm thinking of dynamical models where you can actually implement the dynamics with various sets of parameters. Okay, the same dynamics. The example I have in mind. I don't know if you know the work of Yves Marder on the stomatogastric ganglion in crab, where there are these nice patterned oscillations, and they've been showing down that if you take different crabs of the same species, they implement the same oscillatory dynamics with even somewhat differently connected neural network. And so basically my question is if you have some pockets in the parameter space which leads to pretty much equivalent dynamics, how will your profile likelihood function look like? Will it because you're screening over the whole parameter space in the optimization, will you see it in your function or not?
Yeah, you will see it.
So it will become a bit more flat. But you will not necessarily see from it that that is the case when these pockets are isolated and far from each other, as opposed to.
Sorry. This situation is exactly this situation. Yeah. So what happens here is, if we look at the profile here, we have one solution here, one solution there. Yeah. So here the parameter is higher, the reaction is fast. Here it is slower. Yeah. And if you look, this is for parameter K1, and if you do the same thing for parameter K2, you get the same picture. Yeah. Where this minimum here then corresponds to the other minimum here, and this to this. So these are exactly the 2 islands you are talking of, yeah, and they are equally well fitted because the values here are the same. Yeah, so this is exactly the case where you have 2 isolated points in could be regions, yeah, then this would be a little bit more, yeah, but that there are 2 really far apart regions in parameter space that produce the same behavior. Yeah, and somehow this has to do something with robustness. Yeah, it just increases your chance to survive. Yeah, if you can be there or there. Yeah, but this is it would exactly look like this.